Well, welcome everyone to the John Curtin School of Medical Research. My name is Riccardo Natoli. I'm the head of the Clear Vision Research Lab here at the John Curtin School and the Associate Director of Research Development at the School of Medicine and Psychology. It's a real privilege to be here for a number of reasons. But first, to be representing a little bit of Retina Australia, who I've actually joined as a director at the start of the year. Retina Australia was one of the first funding agencies to actually ever give me any funding and seed funding. And we're able to use some of that money actually to turn it into a successful NHMRC grant. So without the support of Retina Australia, we wouldn't have had any of that support. And also at the local area, people such as Jan James, who did a remarkable job with the local Retina Australia community in the ACT, really helped support the inception of Clear Vision Research. So before I begin, I just wanted to really thank you and thank the patients and the community for all the support that they've showed our research over the last 10 years. So thank you very much. And this is an attempt to sort of reinvigorate a lot of the stuff that was being done in the local community, and largely driven by Julia Hall, who will be chairing this session today, who is the new CEO of Retina Australia. One last thing, I just want to tell you a little bit about John Curtin and what we're doing here. So the John Curtin School of Medical Research, it's a wonderful research environment where we bring together different disciplines, including immunology and genomics and RNA biology and neuroscience together to solve some really, really complex problems. Please come on in, guys. And so having those collection of researchers coming from different areas to address really complex problems is vitally important. And we can only do that through concerted effort of research working together. And with that, I'd like to introduce as well Thomas Priest, who's the Centre Director for the Shine-Dorgano Centre for RNA Innovation, who's working alongside groups such as Retina Australia to bring new therapeutics in the RNA space to market. With all that said, I'd like to welcome you here. I'd like to also begin by saying I'd like to acknowledge the traditional lands on which we meet and pay our respects to the elders, past, present, and emerging. With that said, I'd like to hand over now to Julia, who will take us through the rest of the session. Thanks everyone for coming.
Thanks so much, Riccardo. I'd also like to extend a very warm welcome to everyone here today. It's so nice to see so many people for a live in-person event, and also to have everybody online as well. Thank you so much for your interest. I'd also like to thank our key sponsor, Apellis. Thank you so much for your support. And our event host today, Clear Vision Research, the John Curtin School of Medical Research, and the Australian National University. We have three speakers today, Dr. Carla Abbott, who will be talking about discovering emerging treatments in geographic atrophy. Our second speaker is Nick Barashev, who will be taking us through his 3-minute thesis on using the message of exercise to prevent blindness. And Dr. Adrian Chuanka, who was our 2023 Research Grant Awardee, and he'll be reporting on his research report. We'll learn about how targeting inflammation can protect against retinal inflammation. After each speaker, we'll have time for some questions. So if you're in the audience here today, just please raise your hand and someone will come around with a microphone and you can ask your question. If you're online, there is a button you can press to raise your hand, or you can just type your question in the chat box. If we don't have time to get to your question today, please feel free to contact our office. You can email at info@retinaaustralia.com.au, and we will follow up with the researcher about your question. To kick off our presentations, I'd like to introduce Dr. Carla Abbott. Dr. Abbott is a Senior Research Fellow at the Centre for Eye Research Australia and the Department of Surgery Ophthalmology at the University of Melbourne. And she's also an optometrist at the Australian College of Optometry. Her current research focuses on identifying new treatment strategies for retinal diseases, including age-related macular degeneration and inherited retinal disease. Dr. Abbott trained as an optometrist and obtained a PhD in vision research at the University of Melbourne prior to completing postdoctoral fellowships at the Australian National Vision Research Institute, the SaveSight Institute, and Divas Eye Institute in the United States. She's been a part of the senior leadership teams of the Bionic Eye Research Unit and the Macular Research Unit at CERA since 2013. Dr. Abbott was awarded Fellowship of the Australian College of Optometry in 2014, the Dr. Hitesh Peshavaraya Award for Outstanding Contribution to CERA by an early to mid-career researcher in 2020, and the Macular Disease Foundation Australia Excellence Award in Research Award in honour of Richard Brill's AM in 2023. Dr. Abbott is also a current appointment on the Optometry Board of Australia. Today, Dr. Abbott will be talking about the emerging treatments in geographic atrophy, a late-stage form of age-related macular degeneration. For the first time, there are now approved treatments available in the USA, and it's expected that these treatments will now be considered by regulators in other jurisdictions, including Australia, in the coming months. This talk will cover diagnosis, the potential new treatments on the horizon, the patients that these treatments are best suited for, and opportunities for clinic to be involved in clinical trials. Let's welcome Carla to the stage.
Hi, everyone. So thank you very much for that very warm welcome, and I'm really pleased to be here and be given this opportunity by the organisers to come and have a chat today. So really keen to get your questions at the end, but I'll try and make sure that I'm speaking for those who aren't able to see the slides clearly as well. So yeah, as introduced, I will be talking mainly about geographic atrophy, which is a late stage of age-related macular degeneration. But first, I would just like to acknowledge the traditional owners of the lands we are all meeting on today and pay my respect to elders past and present. And I'd also like to extend that respect to any traditional owners joining us here today as well. Before we start, just a couple of disclosures and acknowledgements. So I did get my flight paid for from Melbourne to attend by Retina Australia today, but I have no other disclosures. And I work within the Macular Research Unit at CERA, which is headed by Professor Robin Geimer, who's a medical retina specialist. And she has also contributed to this presentation and has a number of disclosures as listed. So first, just a little bit of the basics about age-related macular degeneration and its clinical classifications. So we know that late-stage age-related macular degeneration, or I'll call it AMD for short, causes central vision loss. So there is an image up there showing what things look like clearly with a normal vision compared to vision with AMD, where you have central vision missing. And in terms of what you see at the back of the eye at the retina, there's a gradation between, or a clinical stage progression between when you have no aging changes at the retina at all to when you start to get early signs of AMD, which to start with, it's very tiny little yellow dots called drusen that start to develop. They get bigger when you get diagnosed as a clinical stage of intermediate AMD, but both the early and intermediate stages don't usually cause a lot of symptoms. And it's not until the late stages where you actually have photoreceptor loss, which are the cells that detect light in the back of the eye, where you actually get that central vision loss. And there's two types of late AMD, both what we call geographic atrophy, or I'll be using GA for short, since that's what we're going to mainly talk about today. And that's the atrophic type, or you also get the neovascular type, which is when there's bleeding in the back there. So both stages. And the geographic atrophy type can also change to the neovascular type as well. So that's just a little bit of an introduction to AMD. And I'll be focusing on the GA dry type today. So although AMD is a leading cause of vision loss, affecting about 1 in 7 people over 50, historically there has been quite limited treatment options to slow the vision loss. So neovascular AMD had first intravitreal anti-VEGF treatments approved way back in 2004 in the US to start with, and then followed through in Australia. And so those treatments have been available now for 20 years. But geographic atrophy, the other late stage, has not had a treatment at all until the first ones were approved in the US last year, which is not actually approved yet in Australia, but we'll be talking a bit more about that today. And in early and intermediate AMD, there's no treatment available currently to prevent people from progressing to the late stages, either neovascular or atrophic disease. So today we're going to be talking about the emerging treatments for geographic atrophy, or GA. So in February last year in the US, the FDA approved pegcetacoplan, or also called Syfovre, as the first treatment to slow down the progression of GA. And 6 months later, in August, they also approved another drug, avacincaptad pegol, as another approach. So these companies will also be applying in other jurisdictions, including Australia. And the update on that at the moment is that in Europe, it's still under evaluation. And in Australia, there hasn't been any announcement yet about either medicine by our regulating system, which is the Therapeutic Goods Administration. So we're waiting eagerly to hear what's going to happen there. And we're going to talk about all this in a bit more detail. There's also several other novel interventions currently in late-stage clinical trials for GA. So what are these new emerging treatments doing? So the most common pathway that's being looked at is the complement pathway and trying to suppress inflammation. So the complement pathway is a group of proteins that are helping the immune system to work. And I do have an image up there of the roundabouts of that system. But essentially, there's strong evidence that are implicating overactivity of the complement pathway in how AMD develops. And hence, there's many trials for GA now that are targeting potential treatments that work on trying to inhibit the complement pathway, so reduce that overactivity that's known. And the most common point of targeting that is by what we call the C3 and C5 inhibition, which we'll talk about in terms of the medications. So we're going to go through some of the clinical trial data firstly that's resulted in the approval of these medications in the US. So firstly, the pegcetacoplan, there was 2 trials called the OAKS and the Derby trials, and these are what's called phase 3 trials, which is the later stage clinical trial development. So there was over 1,200 participants with GA across 200 sites internationally that looked at this pegcetacoplan intervention. And this is an anti-C3 intervention, and it's using intravitreal injections, either monthly or every other month. So that's injections into the eye itself to try and reduce the growth of the atrophic lesions. So there is an image here that's trying to point out that the macula is quite a large area at the back of the eye in the retina, and the fovea, what we call the fovea, is the very center of the macula, which has the absolute finest vision, responsible for our absolute central vision. So, and you can see there's black areas in there, in that image, that are indicating the areas where the photoreceptors are lost, or we call it atrophic area. And so that's where there's no vision. So these studies were looking at the 12 and the 24-month change in the total area of these GA lesions to see if the drug was able to slow down the growth of the lesions. And this trials also looked at a sub-analysis of the lesion area change away from the fovea as well. So this is called extrafoveal lesions compared to lesions which are under the fovea called subfoveal lesions. And as I mentioned, the fovea is that very central part of the macula. And the trial has also been extended out to 60 plus months ongoing, but we'll show you the 12 and 24 month data now. So for the OAKS study, this is the 12-month lesion area data in graph format here. And for the whole data set, so both the subfoveal and what we call the extrafoveal lesions, there was a 22% reduction in the size in the lesion growth compared to the people that didn't get any of the drug. And that's for the monthly injections. And for the every other month injections, there was a 16% reduction in the growth of the lesions. And then very interestingly, when we looked at the pre-specified analysis of the extrafoveal lesions, so the ones outside of the fovea, there was a 35% reduction in the growth of the lesions in the monthly group and a 21% in the every other month. So it seems to work a bit better for the to slow down the extrafoveal lesions, but I will come back to this a little bit later as well. In the Derby study, again, looking at both all the data all together, there was a 12% reduction in those that are receiving monthly injections, and 11% reduction in those receiving every other month injections. And then when they looked just at the extrafoveal lesions, there was a bit higher rates, so 25% reduction in the every other month and a 16% reduction in the monthly injections. When we're looking at both the Oaks and the Derby studies at 24 months, you see a continuation in this trend. So there was a 22% monthly reduction in those receiving the monthly injections, 18% in the every other month for Oaks, and similar numbers, 19% reduction in the monthly group for Derby and a 16% in the every other month for Derby. So just onto the other drug as well that's now been approved in the US, the Avastin-capsid pegol. So this was a study with 448 participants internationally, and they were getting intravitreal injections as well, and looked at the 12-month change in the total area of the lesions. So the data, you can see a similar trend line where you get a 14% reduction in those that are receiving the monthly injections compared to people not receiving the injections with one statistical method. And they had another statistical method that showed very similar outcomes, 17% reduction. Now, they did not have an every other month option. They just had a monthly injection option. This trial is also being extended up to 42 months ongoing. And they did not have an evaluation of the extrafoveal versus subfoveal data because anybody that had a subfoveal lesion was actually excluded from the study to start with. So a little bit of differences in the study design. In terms of, so that's the efficacy data, how well it works. In terms of the adverse events data or the safety of these medicines, both the Oaks and Derby compared to the GATHER2 studies were similar. So there's very small numbers of adverse events that are similar to other intravitreal injection studies. Of course, you're putting a needle into the eye, so there is a small risk, very tiny risk of infection and so forth, just because that's the approach. The main thing to note is there was seen an increased prevalence of the neovascular type of AMD onset in the treated groups compared to those that did not get treated. And that was a 12% onset in those receiving monthly intravitreal injections, 7% in those receiving every other month compared to 3% that got a sham injection for the Oaks and Derby studies. And in the GATHER2 study, there was a 7% in those that were treated compared to 4% in the sham. Now, all these people were then treated with the anti-VEGF injections, which is the common treatment for those with the wet type of AMD or neovascular AMD. So it was just a matter of treating those people with that. And as I said, there is a small risk anyway of going from geographic atrophy to neovascular AMD. The other thing of note that was actually not found in the trials, presumably because the cases are so low, but is under ongoing investigation, is the American Society of Retina Specialists reported 13 cases from real-world data of retinal vasculitis within 14 days of injection that was not found in the trials. And there's also been one case that was reported as well using the Avastin-capsid pegol drug. And so there's ongoing investigation into that. There's no cause that has been completely clarified at this time, and the exact rate of cases is unknown, but it's very tiny. However, because this vasculitis can have a severe visual impact, so you can have great loss of vision, this risk needs to be explained to patients at consent. Just comparing the two studies a little bit more, the Oaks and Derby to the GATHER2. So they did have the same inclusion criteria for the lesion size at baseline. So the same people going into the studies with the same types of lesion size. But they did have a different inclusion criteria for vision. So the Oaks and Derby participants had to have what we call 6/9 or better vision, which is up to 3 lines of central vision loss on the chart. Whereas the GATHER2 could actually be up to what we would call 695 vision, which is actually greater than legal blindness in this country. So there was quite a difference in who was included in terms of their vision. And then for the lesion location, there was also a different criteria, as we've already talked about a little. So the Oaks and Derby participants could have the subfoveal or extrafoveal lesions, while GATHER2 could only have the extrafoveal lesions. So they actually excluded anyone with the subfoveal lesions. And so the implication is, because over half the eyes in the Oaks and Derby studies had subfoveal lesions, and these generally have a slower growth rate than the extrafoveal lesions, it's unsurprising that they had a slower reduction in lesion growth compared to the sham group, as the sham group will also have a lower rate of growth in itself, just to do with the underlying known how those lesions change. And the other thing that was different between the studies was the criteria for neovascular AMD in the fellow eye, or the other eye. So the Oaks and Derby studies did allow people with neovascular AMD in their fellow eye to be enrolled, and the GATHER2 did not. So this means that the risk of neovascular AMD development is different between the studies, because it's known that people with neovascular AMD in the fellow eye are at higher risk of developing neovascular AMD in their other eye already. So just the key messages on this. So the monthly or every other month injections of both of these medications have been shown to decrease the rate of GA lesion growth, especially for extrafoveal lesions. Overall, the medicines are generally well tolerated. However, there was an increased rate of neovascular AMD, which can be managed, and there are ongoing investigations into post-study reports of retinal vasculitis, which is in very small numbers, and we still need to understand more. It's important for patients to note that there was no noticeable day-to-day effects of visual function, and that patients won't notice an improvement in vision when you get these injections, which is quite different to the neovascular AMD, where you do get this improvement in your vision after you receive the injection. So I'll talk a little bit more in a moment about what this means in terms of clinical management, but it's just important to understand that. And of course, there's ongoing clinical trials to assess the longer-term safety and efficacy of the medications for longer periods of time. So just to recap the regulatory side, so the regulatory approvals for these were obtained in 2023 in the US. Both the European Medicines Authority and the Therapeutic Goods Administration in Australia are still considering their decisions, so it's not currently available in Australia. I'd just like to very briefly touch upon other emerging interventions in advanced stage clinical trials for GA. So this is a table I've got up here that was published in Retina today in November. And basically it's just to indicate there's a lot of studies happening in late stage clinical trials. And there's also a range of targets and delivery methods that are being looked at, including oral, subcutaneous, so under the skin, or also one-off subretinal approaches, meaning under the retina. However, most are still targeting this complement pathway that we talked about at the start. I'm just going to very quickly touch on two of those to give you a flavour. So the beauty of potential subcutaneous injections is there's potential for self-administration, so not having to go into the ophthalmologist's office every month to get your eye injection. So this one is under the Golden Phase 2 trial, which is looking at an inhibitor of complement factor B targeting the liver, because the liver is responsible for the production of that complement factor. It would be able to provide simultaneous treatment for bilateral GA, so you wouldn't have to get injections in both eyes, you just need an injection under the skin. It has over 300 participants and they get their subcutaneous injections every 4 weeks. This is still under trial, meant to be finished this year. So there's no regulatory approvals for this at the moment. The other one to highlight is gene and cell therapies, which have a potential for a once-off treatment. So none of this every month having to do anything. You get one treatment once, and then that's it. So the thing with this is it does require a subretinal injection, which means under the retina. So you need a vitreoretinal surgeon and an operating theatre, but then you only need that once. So what are these for? So gene therapies are to modify a gene that's not functioning properly. And it's basically bringing in a viral vector to bring in a healthy copy of the gene to act on this complement pathway, for example. Cell-based therapies are aimed at replacing the retinal pigment epithelium, which is an important layer below the retina known to be affected in AMD. And this can be applied as a suspension or otherwise on sheets or a scaffold. And it can also, it's made using stem cell technology, either human embryonic stem cells or otherwise induced pluripotent stem cells. And these are, of course, talks in their own right with a lot of detail about how all this works. But just to give you a flavour, the trials for this is due for completion in 2029. There is later stage trials happening internationally, but at the moment, there's no regulatory approval anywhere. So a little bit on how to get involved in clinical trials if you're interested. Firstly, why? Why would we do this? So I guess advantages are the potential to receive a successful intervention, knowledge that you're contributing to the finding of interventions or improving the understanding of eye disease, and also access to a cutting-edge research team with regular reports sent back to your primary eye care practitioner. However, the points to keep in mind, because trials aren't for everyone, the randomized nature of trials mean that you don't know and your treating researchers don't know because it's masked if you're in a group receiving interventions or not. So you have to be prepared that you might not be getting the intervention. And often the way a lot of clinical trials are at the moment, and again, there may be sort of changes in this, is that at the moment you have to usually get into a central location such as an eye hospital regularly, which involves regular travel. At the Centre for Eye Research Australia, there's currently 13 trials on offer for AMD, and I've sort of just listed them all there. The trials are for all stages of AMD, and sometimes the participation just involves a one-off research visit up to interventional studies requiring 2 or 3 years of visits. But clinical care is always maintained with your referring practitioner, and letters are sent back. You can directly register your interest for a trial on our website. So there's a big button there saying register your interest when you go out, and it just takes you through a guided process of filling in an online form. So it's quite easy to do that if you're interested. Of course, you can also talk to your optometrist or ophthalmologist if you're interested to register for trials. And there's usually trials available in each major capital city. In terms of the changes in clinical practice, so what are the implications of these emerging treatments becoming available for GA going to have on clinical practice? So it really is a dawn of a new era, because this is really the first time there's ever been treatments available for GA, which is incredibly exciting. And ophthalmologists and optometrists in Australia, even though it's not approved yet, have been preparing new clinical guidelines for GA when the treatments become available. So RANZCO, which is the governing body of ophthalmology, have just published their new referral pathway last month online, and the optometrists one is about to be released very shortly as well. So that's fantastic. It's important to still recognise that the treatment is not suitable for everybody, and that it needs to be talked through on an individual basis to work out, is this something that's going to be right for this person or not? The clinician will need to be considering the progression risk factors for that person, the treatment risk factors for that person, and the treatment burden, including that at the moment at least, it involves lifelong monthly intravitreal injections. And also the fact that the patient, just to look after their expectations, it's going to reduce the amount of vision loss due to GA over time, but it's not going to cause a day-to-day noticeable difference in the vision. So the patient needs to be prepared for that, because that's different to wet AMD, for example. Imaging of the retina over time is going to be key to clinical decision-making. So it's important to identify those at high risk of progression from early stage AMD to GA. And our lab's done a lot of work in this space, identifying biomarkers. And also it's going to be important to those with GA lesions to work out who's going to be most likely to progress to underneath the fovea and cause that foveal vision loss, which is your really central vision. And as I've already said, there's a great importance on individual counselling on the risks and benefits and talking it through with each patient. So just to sort of show you how we would look at the rate, the risk of lesion growth over time. So the blackness that you can see in these images of the retina is the bits with no vision, the parts that have atrophy. And then you can look in different patients. So we've got a patient up the top with extrafoveal lesions starting outside of the fovea, how it changes from year 1 to year 2 to year 3, and the risk that it might come in underneath the fovea, versus someone down the bottom there who's had a subfoveal GA and how that changes over time from each year. So this is the type of equipment that we'll be using to make those decisions and to talk with patients about the risks and benefits. Okay, last slide, take-home messages. So there's currently 2 proven interventions to slow the progression of GA that are now approved and are freely available in the US. There's many more trials of novel interventions for GA underway. Those targeting this complement pathway are the most numerous and in later stage clinical trials. And these trials are international and are available in Australia, including where I am at CERA. And whilst the frequent intravitreal injections are the mode of delivery of many new interventions, other routes are also being explored, which is really exciting. And you can rest assured that your clinicians, the optometrists and ophthalmologists in Australia are preparing for this time when GA treatments become available here. And we are recognising that conversations with our patients about individual treatment potential and risks is going to be very important. So that's really what I wanted to share with you today. And of course, I'm happy to take any questions. Thank you. Well, yeah. So the question is, what is the uptake of these new drugs by patients in the US? I'm not really, I mean, I guess because it's real world data, it's very hard. I mean, so we're getting some real world studies starting to come through, but I guess I wouldn't have the answer for that necessarily offhand. I'm not sure if would you have more information about that, Mary? Thanks, Mary. I'll just repeat a little bit of that for those online. So there's been 250,000 people being choosing to uptake the pegcetacoplan in the US since it's been available since February 2023, and that the rates of compliance and so forth has been quite high in the trials, and also most people are opting for a 6 to 8 week in conjunction with their clinicians injections rather than every month. So about 70% of people are using the 6 to 8 week regimen rather than monthly. I think I got all that right. Yep. So that's, there we go. That was Mary from Apellis giving you the information from directly. Yes, just, yep.
Dina, yep.
Yep.
I'll just speak up a bit. Just a nuts and bolts kind of question. You've got this current practice in the US. Looking here, when it finally gets TGA approval here, what do you expect the cost will be? Will it be a PBS kind of product, or will it be this living cost?
Yeah. Do you want to just pop down and speak into the, yeah. Do you mind? Just because, yes. Well, because no, there's people on, there's another doubling of the audience online, if you don't mind.
So I'll just repeat the question. The question was basically, once it's actually approved in Australia, this new drug, what is the cost to patients going to be? Is it going to be on the PBS or is it going to be user pay?
Yes, so the company has plans to seek reimbursement through the PBS. So we plan to submit next year, following from getting regulatory approval by the Therapeutics Goods Administration, sorry, TGA.
In the US, what's the position?
In the US, there's a different market, so they have a completely different model. So in Australia, we have a universal healthcare system, whereby the majority of therapies are hopefully funded and available on PBS, and patients pay copay. In the US, they're largely funded by private health insurers. There is a different kind of a fund called Medicare, which is completely different to Australia. It's not the same universal type of an arrangement. So it's a completely different healthcare system in terms of paying for therapies. So patients usually have to have private health insurance if they're not able to afford therapies out of pocket.
Should have started by saying it sounds fantastic.
No, not at all. I hope that helps. Thank you.
Probably have time for one more question if there's one more question. That.
Yeah.
While you come down, so essentially the question from the audience was, why was it approved in the FDA and not approved in the European market?
It's a good question, but each country has its own regulatory body, as you know. So FDA in the US, EMA in Europe. In Australia, it's the TGA. These countries do conduct their regulatory evaluation of the submission in different ways. They are independent, but they do look to what's happening in the rest of the world. So, for example, Australia will be keen, the Department of Health in Australia will be very interested in what's happening in Europe, but they operate completely independently, make their own decisions as to whether or not a product, they consider a product to be efficacious, safe, and they also look at quality as well from a regulatory perspective. Thank you.
Just in the interest of time, we might move on to the next speaker and thank Carla for a wonderful presentation.
Thank you, Carla. That was a great presentation. Next up, we have a special short presentation by Nick Barashev. Nick is a PhD student at the Clear Vision Research Lab here at the John Curtin School of Medical Research. After completing his bachelor's degree in biomedical science from the University of Canberra in 2019, he moved on to an advanced master's degree in neuroscience at the ANU, graduating with first-class honours in 2021. Nick's research at Clear Vision Research is focused on identifying and explaining how the act of physical exercise conveys protective messages to the retina, and to see if we can harness these for the treatment of retinal degenerations. Last year, Nick presented his PhD research in the 3-minute thesis competition entitled Using the Message of Exercise to Prevent Blindness. In this competition, you must present your PhD research in under 3 minutes to a non-expert audience with only a single static PowerPoint slide to support your talk. Nick ended up placing second at the ANU Grand Final. Let's bring on Nick to hear his 3-minute presentation.
Thank you so much, Julia. It's always fun getting the opportunity to present this presentation without a timer that's ticking down, and then so it's always good. So it may go over 3 minutes. If someone's timing it, don't yell at me, please. So for my 3-minute talk, this is the PowerPoint slide that I chose, and it'll make sense in just a second. So when I was 15 years old, I remember waking up in hospital, not being able to see anything out of the bottom left of my vision. I had a stroke, and because of that, my vision now looks like this. Now I want you to just imagine that that blind spot is actually in the center of that image, because that's what people see every day when they suffer with a devastating disease called age-related macular degeneration, or AMD. AMD is caused by damage to the important piece of tissue that lines the back of your eyes called your retina, which is responsible for your vision. But this damage only worsens with age, and even worse, the particular part of your retina that actually damages is responsible for pretty much all your useful vision, including your central vision. So when you suffer with AMD, the center of everything you see disappears. As the leading cause of blindness in Australia, AMD will affect 1 in 7 people in their lifetime, meaning at least one person in each row here today will eventually lose their central vision. What if we could stop this from happening to you using something as easy as exercise? I mean, we all know how good exercise is for us. Going to the gym makes you feel stronger, running makes you feel fitter. But despite these physical benefits, the precise mechanisms as to how exercise protects our bodies, yet alone our eyes, from disease, especially AMD, are still not understood. We've shown that just 4 weeks of physical exercise can actually protect your eyes against the same damage that causes AMD. My research has found that when you exercise, your body produces small molecules called extracellular vesicles, which act like molecular text messages inside your bodies. Not only does your body already use these messages as a normal way of talking to one another, but it's only when you exercise that the messages that are sent to your eyes are changing what they are saying, and that's what's protecting your retina from damage. When we took a handful of those messages from an animal that had exercised and put them into the eye of an animal that didn't exercise, that animal showed almost the same protection to damage as the exercised animal. So can we use this molecule as the message of exercise and practically prescribe it to someone for whom exercise isn't an option? Imagine being able to use your body's natural response to exercise, using what's already inside our bodies to stop AMD and stop you from losing your eyesight later in life. Now, I want you to take a moment to look at the image on the screen and just imagine waking up every day and seeing that. Because of my stroke, this is what I see when I look at this memory of me and my beautiful mum. This is what fuels my passion for finding a cure for AMD, because if we don't do something now, for 1 in 7 of you, that blind spot will be in the center of that image. Thank you. Are there any questions?
What sort of exercise then?
So the model that we've shown protective effects of exercise is just aerobic running, just voluntary exercise. Yes.
So Nick, you said that if you could recreate those messages
Yeah.
for people who can't exercise,
Yeah.
how would you do that?
Well, if we can find out what's inside those messages, when we took messages from animals that had exercised, and then we took the same messages from animals that didn't exercise, and we saw a functional difference in the degree to which they protected the retina in between those two groups. So if we could find out what's actually inside those extracellular vesicles, find out what RNAs, what lipids, what proteins are inside them, what's stopping us from making our own message of exercise and giving it to someone who physically can't? And so the extracellular vesicles, the contents are different to what's inside the sedentary ones. We've tried to extend the experiment. So what we actually tried to do is we tried to eliminate these extracellular vesicles altogether. But we saw some interesting findings. So it's still on the cards of further experiments that need done.
There's 2 wonderful questions online, so I'll just try and repeat them. The first one is, Nick, how much exercise?
Yeah. So how much exercise? How much exercise needs to be different? So we've shown that 4 weeks of exercise provides functional protection to the retina following the damage that causes AMD. But 2 weeks of exercise didn't actually provide that degree of functional protection. We did start to see molecular changes in the retina that hinted at changing levels of inflammation in the retina, but 2 weeks didn't provide that level of functional protection. But 4 weeks did. I'm very curious to see whether 8 weeks or 16 weeks or a year's worth of exercise would be enough to provide that level of functional protection.
And the second question was, what are extracellular vesicles?
So extracellular vesicles are, as I mentioned in my talk, they're like molecular text messages inside your body. So every cell in your body normally uses them to talk to one another, exchanging information. So the cell's like, I need this, give it to me, I'll give you this back. It's another way, it's a way that your cells use to talk to one another. They're like text messages inside your body, and your cells use them normally. But when you exercise, the number of these messages actually increases by 2-3-fold. And we think that's what's inside them that's conferring the beneficial effects of exercise.
I think there was one more question from Daisy.
Thank you.
So how do you plan to administer
So when we,
Repeat the question.
so the question was, how do we administer these extracellular vesicles? So what we did to show the protective effects of these exercised extracellular vesicles was we exercised some animals for 4 weeks, and then we didn't exercise some animals for another 4 weeks. And we took the EVs from just their blood, so just a systemic response of these EVs. We isolated them by spinning them very fast, and we put them straight into the eye of an animal that didn't exercise, and then we exposed them to a damage paradigm that mimics the effects of AMD. And we saw that the exercise EVs actually were more protective than the sedentary EVs. So that leads us to think it's what's inside those EVs that's what's conferring the molecular and beneficial effects of exercise. Does that answer your question? Yes, intravitreal injection, so straight into the eye. Ideally, it would be the dream scenario to give like an oral administration of these EVs, and somehow they make their way to the eye, and that would be the dream goal. We do have a question from Ryan at the top.
I was just gonna say, with the exercise, obviously, 5 to 4 weeks, do you think how long someone can go to form of your exercise? Do you have like a, you just know how long that athlete, so is there a problem with like, you don't have to be released,
Yeah.
so just speaking to that.
So the question was, when we measure the amount of exercise over 2-4 weeks, do we have certain animals that don't exercise as enough? How do we quantify the amount of exercise? Because we do these in animals, we have a bunch of running data metrics that we have. It is a voluntary exercise paradigm, so we're not actually forcing these animals to run. And they tend to do a lot of their running at night. And overnight, these mice run, I've seen a mouse run 38 kilometers in one day. Some of them run a lot more than other mice, but they run from like 10 to 20 to even 38 kilometers in one day. So over 4 weeks, they run 4 or 5 marathons worth of running. But what we've actually found from some interesting data we have is that it's not actually the distance that is a measure as to the degree of protection that the mice will have. It's actually the amount of times that the animals engage in exercise that is the most influential factor on what's happening in the eye.
What cells are they using when you exercise? Is it muscle cells or is it other cells?
That's what I would love to know that, because a key drawback with the research of extracellular vesicles is cell surface markers that identify which cells these EVs are coming from. Because all the cells in your body, like every cell, produces EVs just normally, it's hard to identify what cells these EVs are coming from. There's been a lot of research hypothesizing they're coming from platelets, red blood cells, muscle cells. There's been some people that have shown that they don't come from muscle cells, but other people saying they do. So I'd love to know that.
Josh, you're right ahead.
Just in terms of like you've tested it in terms of protection for the eye, is there any other effects that you noticed on other parts of the body? For instance, like did you mimic the fact that someone hadn't done exercise or did they get the benefit of doing exercise by injecting it?
So
could you basically send the message that you had done exercise and suddenly get the fast
that's a very common question that comes up with this the EV story is, could you get the same sort of physical benefits of exercise by injecting the EVs? Not that I know of. We were obviously taking the systemic response of exercise from the blood EVs and putting them straight back into the eye. The effect we were hoping to see was just the less neurodegeneration. We wouldn't see any other sort of physical effects by injecting them into the eyes. But no, I'm unaware of people taking EVs, injecting them, like from athletes, and seeing if they run faster. It would be interesting. We do have a question online, I believe. Is there research to show that people who exercise more have less incidence of AOD? Yes, there are a few population studies that have been done that have shown that people who report higher degrees of physical activity are less likely to develop AOD. But there are limitations in reporting of physical activity because not many people want people to know that they're lazy.
Sorry, it's a follow-up. The number of people in Israel, there are 7.
Yes.
So I'm a little worried. I'm also worried that I'm not capable of running 46 kilometers overnight. You know, what sort of, when you said running, are there other exercises that you can do?
We believe that you can do it. Come on.
You do.
That's it.
So it's a good question. Are there other forms of exercise? There is a lot of research into which form of exercise provides the highest degree of protection, but unfortunately, no one really looks at the eyes in that aspect. Usually it's to do with memory loss, motor impairments, and all that other sort of stuff. But there is a lot of research going into high intensity interval training, which correlates well with our running data. Shows the number of times you engage in exercise. It's not actually the degree or like the amount or the load of exercise that you're actually doing. Obviously, like strength athletes will be a lot stronger but less fit than a running athlete. But will they have the same level of protection in the eye? Will they have lower incidences of developing AMD? I'm not sure on that aspect. But the data we have goes to show it's voluntary exercise. So just running. And whether 20, 30 kilometers in a mouse translatable to a human, that's like an ultra marathon a day. Keeps the blindness away. Yeah.
Look, if there aren't any further questions, can you please help me in thanking Nick for a wonderful presentation?
Thank you so much, Nick. We'll all start our marathon training tomorrow, I'm sure. Our final presentation today is by Dr. Adrian Chuanka. Adrian is a postdoctoral fellow here at the Clear Vision Research Lab at the John Curtin School of Medical Research. His research is centred on the development of new treatments using RNA to target certain genes involved in retinal degenerations. Adrian's experience in this area stems from training in molecular biology during his PhD. The retina is the part of the eye responsible for capturing images and sending them to the brain. His thesis explored why and how it starts to deteriorate or fail by closely examining the activity of many genes at once. Adrian was awarded the Frank Fenner Medal for the most outstanding PhD thesis submitted in the John Curtin School of Medical Research for his PhD thesis in 2022. Adrian is also involved in the establishment of EthroSite, a biotech venture emerging from the Clear Vision Research Group. He teaches the genetics and cell biology component of the ANU medical program and lectures on retinal physiology within neuroscience courses offered by the Eclectis Institute of Neuroscience here at the John Curtin School of Medical Research. In 2023, Adrian was awarded with a research grant from Retina Australia for the project using RNA silencing to tackle neuroinflammation in retinal degeneration. We'll now hear about the results and outcomes of this project and learn about how targeting inflammation can protect against retinal degeneration. Thank you, Adrian.
Well, thank you for the introduction, Julia, and thank you for the kind words. So really, today's presentation is trying to describe to you guys how we spend the money that we've got from Retina Australia and what research we've done in this project. So the key area of research that we focused on was inflammation, but specifically, how can we target inflammation to actually slow down retinal degeneration? And what I'll explain at the beginning of this presentation is how inflammation is really embedded in the context of AMD pathogenesis. Just move the monitor so I can see that side. So the motivation for doing this work is very simple. It really can be summarized in 4 facts that I've got on this slide. 200 million people right now are affected by AMD. 1.5 million of them are currently in Australia. And here we have a couple of patients that have attended this presentation. So really we welcome you. And it's always amazing to actually be able to hear the patient's perspective and what it's like to live with a disease, because that really motivates us to do this work, but it also enables us to probably optimize our research and really build better therapeutics. In fact, 17% of Australians over the age of 50 experience vision loss as a result of age-related macular degeneration. And we've heard all about what really happens in the retina in the previous presentation when someone has AMD. We've also heard that there are 2 drugs now that are approved for AMD. And it's amazing to actually be able to see that, patients do have access to drugs right now, but also as it is with any drug, there is always room for improvement. There is science to be done to be able to improve how well these drugs actually work. Now, the main topic for today is really inflammation. And there are many definitions for inflammation. And, maybe some of you may be familiar with the idea that, if you fall and you have a scratch or something like that, that area which it is damaged becomes inflamed. If we look at the definition of inflammation, it's just take it from the dictionary, is a biological response of body tissues to harmful stimuli, such as pathogens, damaged cells, or irritants. So for today, really, when we talk about inflammation, is the retinal response to damaged cells. So again, just to sort of make that very clear, when I refer to inflammation, is what happens in the retina when certain cells within the retina are damaged. So there are many, many reasons why cells can be damaged in the retina. We've heard about some of the factors that may cause AMD, and really we don't have to focus on exactly what these terms mean here, but things such as oxidative stress, there is accumulation of drusen, these are lipids within the eye, they can accumulate, certain cells within the eye may not function properly. There are changes to the vasculature within the eye, so blood vessels can change. And a lot of those elements that occur within the eye, many which are actually stemming from aging and aging processes, a lot of those can actually cause cell damage. And what the retina does when there is damage, it begins to respond to the damage. And one of the responses that occurs within the retina is inflammation. So what inflammation is supposed to do is actually attenuate or resolve some of the cell damage which happens within the eye. The problem is that for many reasons that we don't really understand, when inflammation happens within the retina, it can actually damage even cells that are healthy, that are undamaged. They can actually be damaged by this inflammatory response. And the rationale for current drugs is that if we can slow down inflammation, then maybe we have a chance to essentially slow down how the retina actually degenerates. So really here's the sort of the main idea. Current therapeutics, what they try to do and what we try to do through our research is slow down inflammation. If we slow down inflammation, then the disease should not progress as quickly. And if it does not progress as quickly, then maybe those patients would actually have their sight saved. So that's sort of the main science that we have to cover today, is just looking at how inflammation happens in the retina and how can we actually slow that down. What you're looking at here is a picture of a mouse retina. And what we do in our lab is really we try to understand when there is inflammation in the retina, what really happens, and what are some of the molecular factors. So what are some of the molecules that really drive inflammation? So what you're looking here on the left-hand side, here you can look at a retina of a healthy mouse. And the mouse retina and the human retina are very much the same. There are some differences, but really they're very comparable. And there's one specific part of the retina that we need to focus on here, which is the photoreceptor layer. The photoreceptor layer sits here in the retina, and the photoreceptor layer is really the cells that actually can respond to light. So light comes into your eye, it passes through your retina, and then eventually it hits the photoreceptor cells. When the photoreceptor cells are detecting light, they send a response to the brain, and that is what vision ultimately is. But these cells are the ones that are actually damaged, along with other cells in age-related macular degeneration. And if you look at this particular layer here, there's one feature that I want you to sort of notice here. You can see that there aren't any of these other red cells within this area. So what the red cells in this picture are called microglia, but these are cells that in the retina drive that inflammation. So when we talk about slowing down inflammation, what we really try to do is slow down how active these microglial cells are and how much damage they can cause to the retina. Okay, so then if we actually cause degeneration in the retina of a mouse, and here what we do in our lab is we use animal models that can have degeneration occurring in the retina. So here what you can see is the retina from an animal that actually has degeneration. And if you look at the same layer here, the photoreceptor layer, which again is the layer that responds to light, this particular layer here actually now has a lot of these immune cells here you can see in red, which infiltrate into the photoreceptor layer. And because they infiltrate in the photoreceptor layer is really why there is a lot of damage done to the photoreceptors. And if you lose photoreceptors, you lose vision. It's that simple. Now, why is it so hard to actually slow down inflammation? The reason why it's very, very difficult to slow down inflammation is because inflammation is a complex biological process. We don't have to go into the details of what happens at the molecular level, but what you're looking at in this picture is the molecules within the retina that we have identified to actually drive inflammation. So within this square here, what we have is every single line here is one molecule or one gene. And if they show up in blue, it means that that particular gene or molecule is really it's not found in the retina at high levels. But if it is in red, it means that its expression goes up. And what we identify in the work that we've done for this year and also previous work in the lab is that there are at least 700 or so molecules or genes that actually can drive inflammation. We've heard a little bit about how drugs for AMD actually work. They target one pathway, and that pathway probably comprises of up to 10 molecules. So current drugs affect, up to, let's say, about 10 molecules that can cause inflammation. We know that there are at least 700 of them that can drive inflammation. So if you want to improve how we actually treat AMD, what we need to do is be able to come up with a therapeutic that actually can target can target a very large amount of proteins or genes that can actually drive inflammation. And this was really the premise of our work. Is there a way that we can target a large subset of these molecules that actually cause degeneration, so we can improve how effective our drugs are in terms of slowing down inflammation? So that's sort of the rationale of our work. Now, there are some ways that you can actually target a large amount of molecules within a biological system. And one way to do it is to actually identify special molecules called transcription factors. So this kind of molecules, what they can do is they can go into a cell and they can actually make their way to the DNA of the cell in the nucleus. And the nucleus is like the brain of the cell, right? So these transcription factors can go into the brain of the cell and essentially orchestrate which genes or molecules are increased or decreased in expression. So what we wanted to do in our work is maybe try to identify a couple of these transcription factors which are known to be linked to inflammation. And if we can identify those, then it gives us a way to actually target a large subset of molecules that can cause inflammation. So that's really the premise of our work. Can we then find some transcription factors that we know that they drive inflammation, and to see if we actually inhibit them or silence them, then would that actually be beneficial to the retina that undergoes degeneration? So that's exactly what we've actually done. Now, before I actually describe the results of the work, I just want to illustrate the power of transcription factors. What can this molecule really do? If you can find the right transcription factors in a biological system that controls a process, you can really reverse that process. And in fact, some people have shown that in the retina of old mice, these are old mice that have lost their vision, the age of retinal cells can actually be reversed if you can find the right transcription factor to use in that process. So in other words, transcription factors, if you know which ones, if you can identify the right ones, can really reverse the age of all cells and then make those cells young again. So this is how powerful transcription factors can be. The challenge is, though, trying to identify the right transcription factor. Now, so because transcription factors can really reverse biological age, we really believe that they can actually slow down inflammation. I would argue that it's a little bit more difficult to reverse age than it is to slow down inflammatory processes. But maybe some immunologists, if we have any in the audience, probably will disagree with me, but we can chat about it. So how did we find these transcription factors? Okay, so what we have the advantage of in our lab and in many, many labs now is we can take a biological system, we can take a retina, and we can basically look at all the molecules that change in expression if there is damage. So what we've done is we've taken our animal model of degeneration, and just to explain a little bit what this animal model is, all we do is we have mice and then we just place them in their regular housing boxes, but there is a bright light in them, and being exposed to light for about 5 days, it does actually cause some damage to the retina, much in the same way that actually our eyes get damaged by light across across our life. So what we've done is we've taken then that retina and we looked at what is this gene expression signature. In other words, what molecules really change in the retina that now has damage. And then we applied some statistical methods and a number of other mathematical tools. Many of my coworkers call them Adrian nerd things. But we've taken that data and then we've done a little bit of maths on that. And then from that maths, we've been able to basically make some predictions on, like, here may be some of the transcription factors, some of these very powerful molecules that may drive inflammation. And then once we have then identified those, then we can work on ways of actually controlling their action, ways to inhibit them. So essentially, just to summarize this pipeline, we looked at, large data, we've applied some statistical methods, and then we found some key regulators of inflammation in the retina. It's that simple. So here what you're looking at is we've identified three of these transcription factors. I won't mention their names because they're horrible and they really don't mean anything, but let's just call them transcription factor 1, 2, and 3. So TF1, TF2, and TF3. And what you're looking at here in these graphs is very simple. It's what is the expression of that molecule in the retina as the retina becomes damaged in our model. So on the x-axis here, what you're looking at is just, the time since damage. So here we've got the time since we initiate the damage. And here you can see that even within 6 hours after damage, we can see that all these 3 molecules are increased in their expression. And if you look all the way down to 5 days, you can see that these molecules are now expressed at very, very high levels. And we knew from our predictive analysis that I summarized in the previous slide, these 3 molecules together, all 3 of them actually can control many other molecules in the retina that drive inflammation. In fact, I said that it's about 700 different genes we identify that can drive inflammation. And really together, these transcription factors can actually together target over half of them. So now we have the potential, therefore, of developing a therapeutic that not only targets 5 or 6 different molecules, but over 300 to 400 of them, which in principle, it should really improve how effective a therapeutic might be in the retina. So with that, now for a particular therapeutic or a drug to work in the retina, it really needs to target the right cell types in the retina. And I won't give a description of, retinal anatomy here. It's really not the purpose of this presentation. The key point is that, as I said before, there are some cells in the retina that drive inflammation. They're called microglia and microglia. The names don't matter. The point is there are specialized cells in the retina that drive inflammation. So after we identify our key molecules, we just look to see, well, which cells are they in, hoping that they are in the cells that drive inflammation. And sure enough, they were. So here what you're looking at is the morphology or the anatomy of the retina. And in blue, we've got the cells which actually have very, very high levels of these transcription factors. So now what do we know? We know that we've got 3 transcription factors that can target hundreds of molecules that can drive inflammation. And those 3 transcription factors are found in the cells that drive inflammation. So we are working towards a potential way to now target cells that drive inflammation in the retina, which really is the purpose or the crux of our work. So, of course, all of our findings were from animal models. And I briefly described what is the animal model that we use to actually drive inflammation and to cause degeneration. But of course, if we want our data to be valid, to be solid, we have to make sure that what we find in animal models really also happens in the retina of people that have AMD. So for that, what we've done is we looked at the gene expression signature, same idea. We looked at all the molecules in the retina of people that have AMD, and this is tissue that comes from people that have donated their eyes for research, and predominantly coming from the SafeSight Institute. And then here what we found is that the very same 3 transcription factors, these 3 powerful molecules, increase in expression in people that have AMD. So the exact findings that we saw in our animal model, also we see in people that have AMD, which really is a great finding to have, because what it means is we can take our animal models and really build on that data to be able to actually develop treatments and be able to test them in animal models before, hopefully one day, many, many years from now, make their hands into the patients that actually need them most. Now, I'll summarize just some of the findings that we've got from this work in terms of whether or not our approach to treatment works. So here what we've done is we had our 3 different molecules that we knew if we inhibit them, we should be able to slow down retinal degeneration. And then what we've done is we made a cocktail of inhibitors that can actually target these 3 molecules. So these are different set of molecules. And if you inject those into the eye, what they're supposed to do is basically slow down how active these transcription factors are. So we have this cocktail of molecules that can target our 3 transcription factors, and we injected those into the eye. Then after that, we had our damage paradigm initiated. So this is where we again expose our mice to bright light, and their retina sustains damage. And then we assess that retina. Basically, we look to see, can these mice actually see better? So in other words, if we take this cocktail of inhibitors and we inject that cocktail of inhibitors into a mouse's eye, does that mouse see better at the end of our degeneration paradigm? And then here's what you're looking at. What you're looking at here is basically the response of the retina to light. And put simply, what we found is that mice that received our injection with our inhibitors could respond to light better. And what we can say about these mice is that they can therefore see better. So really this shows that, our therapeutic at least has the potential to slow down how degeneration occurs in the retina and potentially allow for now mice to see better, but hopefully in the future patients to see better. We've also looked at another key factor here. So I talked about that this approach to treatment really is a way to reduce inflammation. So for that, we also wanted to make sure that if we look at how much inflammation there is in the retina in response to our treatment, that should be reduced. And that's what you're looking at here. Here is the retina of a mouse that did not receive the drug and that received the drug. And remember initially in my second slide, I talked about like how these immune cells come into the outer retina here, into the photoreceptor layer, and their presence there really causes a lot of damage. So what we've done next is very, very simple. We said, well, okay, if we inject our drug into the eye, are there less immune cells into the photoreceptor layer where they shouldn't be? And that's what we found. Here you can see a large number of these immune cells found in the area of the retina that's undergoing degeneration. And if you look at the animals that received the treatment, we did not see that many cells. So what this indicates is that our approach to treatment really slows down inflammation, and it prevents immune cells making their way to the photoreceptor layer where they can cause damage and therefore destroy the photoreceptors, and then basically that leads to blindness. On the right-hand side, you're just looking at the analysis of the data, and just to summarize it very simply, if we inject our cocktail of drugs into the eye, not only the animals can see better, but they have less inflammation. And we've done a number of other experiments to make sure that this actually works. I won't go through all of that, but the point of this presentation was to summarize what we have done this year, which is that our cocktail of inhibitors that we know which molecules they target can slow down inflammation, which therefore can preserve sight. But there's one other sort of point that I want to touch on. Doing the research in the lab is amazing. We can actually, inform the community on how to, for example, target inflammation. But if you want to get a drug from the lab into the patient's hands, we need to do much more than the research. And really what we need to see is that there is interest in this kind of therapeutic from actually large corporations such as Apellis, but really any other large pharmaceutical company. Because to get a drug to market, it's not cheap. It requires a lot of work, many, many millions of dollars, and it is a long and arduous journey. And what appears to happen in the market right now is that many large companies such as Pfizer, Eli Lilly, Novartis, they really try to invest into this idea of like using transcription factors as the next generation therapeutics for many, many diseases. They're not really working on age-related macular degeneration. They mainly are focusing on cancer, because in cancer, if you identify the right transcription factors, you can prevent cancer. But there is a shift in using the same approach that we have worked on for the last 2 years. And then because we see interest from other companies working in this area, maybe this might be just the right time to actually try to form new partnerships and really try to get something like this to market. And it's amazing to be able to have it in one room today, researchers, patients, and also representatives of biotech companies. This is the kind of event that we have to have more often to be able to actually expedite the development of drugs and also bring transformative drugs to market so the patients can actually benefit from them. So that's all I've got for now, but I'm happy to take on questions.
Thanks, Adrian. Please, Peter.
Yes.
I know David Sinclair over in the States has been doing work on biological clock. Can I just...
That was the work I presented, yes.
Well, that's really my question. I mean, were you actually saying that old blind mice could regain their sight?
Yes, that's exactly what they found. So they had a specific model, which is a glaucoma model, which affects a retinal ganglion cell within the eye. And what they found is that on the surface, the ganglion cells appear to be dead, not functioning. But when they injected 3 transcription factors, those cells became functional, and they looked at their biological age. So there are ways that you can actually say how old a cell is, but just by looking at its DNA. And what they found is that cocktail of transcription factors essentially not only made those cells functional again, but now they looked more like cells from a younger animal. They've actually taken that to a next level where they basically had very old mice exercising, running, similar to what Nick presented. And then after tuning the activity of specific transcription factors, the old mice ran way more than the younger mice. And I think one day David Sinclair would probably turn himself back into a baby. He needs to be really careful. Like, he just looks much younger than he used to. I don't know what he's doing, but we want some of that.
Does this work if the cell eventually dies?
So I guess if you lose the cell, there's probably no way to bring them back. But in diseases such as AMD, you've got, let's say, many like photoreceptors and RP cells that are still there, but they appear to not be functional. So long as the cells are not gone and completely atrophied, this might be a way to actually kind of like reinvigorate them, breathe life into them, if you will.
Any further questions from the audience or online? Daisy?
So
Please repeat the question.
yeah, the question was whether or not these transcription factors have different levels of expression in different types of AMD, such as early AMD, intermediate, and then late stage geographic atrophy or neovascular AMD. The answer to that is when we actually look in samples from like early AMD, intermediate, and late. And then what we found was that even in patients with the earliest form of disease, actually had heightened levels of these transcription factors. Not only were these transcription factors very highly elevated in the retina, they also found at high levels systemically. And actually one of them was one of the best predictors of early AMD. So I guess to summarize that, they appear to be switched on early in disease and then stay switched on all the way to the latest stage of disease. We haven't looked into neovascular AMD. We've only looked at early, intermediate, and geographic atrophy. My prediction is that they probably found at high levels in neovascular AMD too. And I guess to add to that, we found them to be highly elevated in the central retina, but also in the periphery. So I guess they're very good markers of like early stress, which probably happens across the entire retina, not just in the macula.
While people are thinking of their questions, I might ask you a quick question and then I'll come to you, Michelle, if that's all right. Yeah, thank you. Thank you. Just so less about the science and more about sort of Retina Australia and what it meant to actually get some funding from Retina Australia to do some of this work. As a young scientist, what did that mean to you?
It was my first large grant that I've got. And it really not only enabled us to do the work, but it's really a good way for me to be able to actually continue that work. And then essentially to show to people that, I can probably drive this research and then use this as a platform for larger funding, which we were planning to apply for this year. A couple of things got in the way at the beginning of the year, but really the plan now is to basically use the data that we've got here and also the expertise that we actually have been able to get with Retina Australia's help, then take this further by applying for larger funding and then probably try to somehow get this treatment to become a real treatment so patients can have access to it.
Michelle, question.
I was wondering, so these switching factors, do you think they might also be possible for inherited generation? So that they're gene related things that maybe
So we have looked in a couple of different other degenerations in retinitis pigmentosa, and then we actually found that they also expressed at very high levels. And also at the same time, I looked at some of the previous funding that Retina Australia has actually provided to, forgot the name of the person that actually got the money, but the molecule they were looking at has just been revealed to be one inhibitor or one of these transcription factors. I think at that time, they were thinking it works in a different way, but at least we've seen like in retinitis pigmentosa and also in glaucoma that we see elevated levels of the same transcription factors. So I think it's just, they're very, very good markers of stress and inflammation. And inflammation essentially is found in any type of degeneration, not just AMD, but the inherited diseases too. Thank you.
Might just add one follow-up question. So Adrian, we're talking about retinal degenerations, but what about neurodegeneration in general? Are there any lessons that we can learn from the eye that could be applied for other diseases like Alzheimer's or Parkinson's?
It's possible. I mean, I'm not, super familiar with the pathophysiology of some of the other ones, but a key feature there, say it again, it's inflammation. And particularly there's inflammation driven by astrocytes. There's astrocytes in the retina too. There's another type of cell in the retina called the Mueller glia, which works similar to an astrocyte. And then we know that these transcription factors, in fact, are found in the exact same cell types that are also found in the brain. They become very, very active in other types of neurodegeneration, such as Alzheimer's and also Parkinson's. We have looked through the literature a little bit, and yes, in some models of Alzheimer's disease, there appears to again be heightened levels of the same molecule. So maybe it seems like they're going to treat everything.
We probably have time for one more question, which has just come online, which is more about AI and the role that AI might actually play in trying to identify new molecules, including potentially these transcription factors. Can you talk a little bit about how AI might be affecting your work?
Well, so with AI, so like, so we've got essentially what we do these days is we take a damaged tissue, and then we can really look at all the molecules that change in that damaged tissue. But it's increasingly difficult to basically take that very large data sets and find a way to then sort of get down to what is really the central element of that data and what should we really target to be able to then control these very large sets of molecules. So I think with the advent of AI, what we probably can do is basically make better predictions as to what are the right transcription factors to really target and potentially be better at guessing what we need to target to be able to develop new treatments. So I think already in cancer that's been happening. And also with that, as we get more and more data available from many patients, so we're not limited by just by animal models, but really get data from thousands and thousands of patients, then using that sort of large data with AI and its predictive power could really inform what molecules to target next. And then if we build the right infrastructure for producing those molecules, which I think it's happening because of COVID-19, I think AI with the sort of investment in infrastructure and then dedication from scientists and patients, we can probably do better.
On that, can you help me thank Adrian for a wonderful presentation? We'll hand back to Julia, who will close out today's session.
Thank you so much, Adrian, and congratulations on your very successful research project. We look forward to how that research progresses and hopefully in the future to future treatment. Thank you to all our speakers today, Carla, Nick and Adrian, such amazingly informative and interesting presentations. I think we can all be very encouraged by the progression of research and increasing our understanding by being here today to hear directly from the researchers, and also encouraged by the fact that there are treatments which are approved overseas and hopefully will come to Australia soon. Thank you to our key sponsor again, Apellis, for your support and really allowing us to hold this in-person event today. And also to Ricardo. Thank you so much, Ricardo, and your amazing team for supporting us to have and hosted this event here today. All the volunteers who've assisted have been absolutely amazing. Thank you to all of you in the room for attending. It's really so lovely to see so many faces. And all of you who are online today, thank you so much for your interest and joining us today. For those who are here, if you would like to take a tour of the research facilities, there will be volunteers just outside that will be happy to take you on your tour right after this close. Thanks so much and good afternoon.